Alright, so this video is going to cover the last two of our macromolecules, and that is going to be our proteins and our nucleic acids. And we're going to start with our proteins. And kind of like carbohydrates, this is a word that we're somewhat familiar with because we hear it a lot when we talk about diet, and especially when people are trying to live a healthy lifestyle, they talk about cutting back on carbs and increasing their protein intake. But proteins, kind of like the carbohydrates, they are polymers. Remember, that means they're made up of repeating units. And in this case, proteins are made up of amino acids, and that's the monomer for proteins. So there's 20 different amino acids that exist. Some of them we have in our bodies naturally, and we make. Other amino acids we can only get through the foods that we eat in our diet. And that's why it's important to eat proteins, is because we need the amino acids so that we can build other proteins that we need in our bodies. And so the structure of the protein is everything. Okay, once again, a very important, just general idea is that structure determines function. And in the case of proteins, the structure is going to come in the form of the amino acid chain. And so it's all about the amino acids, and that's going to determine how they twist and fold and take on a certain structure or shape. Now, it's kind of easy to define some of our macromolecules, like carbs or sugars that give us energy, lipids or fats that can store energy. With proteins, it's kind of hard to summarize them just because there's so many different types and there's so many different functions. There's a lot of different roles of proteins in our cells and in our bodies. And so here we just have a few that are listed. We have structural proteins that make up a lot of our body structures, things like muscles. You've got actin and myosin that help our muscles contract. You've got proteins in your skin like keratin. You've got proteins that make up your bone structures. They can be very important in regulating cell processes. Some of your hormones are proteins. Now, some of your hormones are lipids, like we talked about the steroids, but some of your hormones are protein hormones. Also, enzymes, we've talked about those in this lesson already. We're going to talk about them again here in a few minutes. There can be transport proteins. When we talk about the cell membrane, they transport molecules into and out of the cell. They can also transport within our bodies, like hemoglobin is a protein that transports oxygen in our blood. And they can also help us fight disease. Antibodies that we make in our immune system, they are going to be proteins. So there's even more functions than what we have listed here, but proteins, there's a lot of different structures and a lot of different functions, and that goes back to those amino acids. So just a little bit more on the amino acids. Okay, every amino acid has the same basic structure. They have a carbon bonded to a hydrogen, bonded to an amino group, and a carboxyl group. Okay, so every single amino acid, all 20, have this same basic structure here. The reason there are 20 different amino acids is because there are 20 different chemicals that could be found here. Okay, there could be an element there, there could be a compound there, but that's what gives each amino acid its own properties. And so when those amino acids join up and they form chains, how those amino acids are going to interact are going to be dependent upon what's right here in the molecule. Okay, so the properties of the amino acids and the order of the amino acids is going to determine the protein shape, which will then determine the protein function. And so here's your 20 different amino acids. You can see that some are nonpolar, some are polar, some carry a charge, and those are all going to affect how those amino acids interact with each other. So there's 4 different levels of structure. Okay, your primary, which means first level, is simply a chain of amino acids called a polypeptide. So it's that basic polymer, amino acid linked to amino acid, linked to amino acid, linked to amino acid. And so that polypeptide is just your basic protein structure. It's a chain of amino acids. Now, proteins do not work in this form. Okay, this is just the start. When we talk about protein synthesis in our cell lesson, essentially this is what we are making, this basic polypeptide or chain of amino acids. Now, what will happen is once that chain is formed, the different chemical properties of the amino acids and those R groups that we talked about it's going to cause that protein to kind of start to twist and fold, and some hydrogen bonds are going to form. And that can lead to either what's called an alpha helix or a beta pleated sheet. And so just due to the interactions of the amino acids, that chain is going to twist and fold in different regions. And then what will happen is now new R groups are exposed to other R groups, and so we're going to have some more interactions. It could be hydrophobic interactions, there could be some ionic bonds that could form, some more hydrogen bonds that could form, and what's going to happen is that protein is going to fold up to form a 3D structure. It's literally sometimes referred to as the globular structure. It doesn't look like it's fancy and specific, but this very intricate folding always goes back to that original chain. If there were different amino acids, it wouldn't take on the exact same shape. So it starts here, it twists and folds, and then it kind of globs up or folds even more to form this intricate folded structure, this 3D structure. Now, some proteins have more than one of these chains, and if that's the case, that's when you would have quaternary structure, or the fourth level, if there were multiple of those protein chains that made up the structure. Like hemoglobin, for instance, has 4 different protein chains that make up that protein. So this is when you have more than one chain. It's still going to be in this highly complexly folded structure that we see here. And this right here, the tertiary structure and quaternary structure, this is when the protein is actually a functioning, working protein. And this shape is super specific, because if this shape changes, then the protein is no longer going to work the way it's supposed to. And so one very important example, once again, we've seen these before, but are enzymes. They're special proteins. They regulate chemical reactions, therefore they regulate our metabolism. Remember, they speed it up, they lower activation energy, it allows the reactions to happen much faster, and so they're very important in keeping all the chemical reactions going in our bodies. Now, they are very sensitive. Homeostasis is critical, because if temperature gets too hot or too cold, if pH becomes too acidic or too basic, what will happen is this protein will literally kind of unravel and lose its shape. And if that protein unfolds, or the word for that is denature, if denaturing occurs and that protein loses its shape, it's not going to work anymore. Okay, and once again, temperature and pH are some of your main contributors to that. So it's very important that we maintain homeostasis, or our enzymes could stop working. And if our enzymes stop working, then those chemical reactions are not going to happen fast enough. Now, a lot of times you can recognize enzymes because they end in ase. Like there's one in your mouth called salivary amylase, and its job is to break down starch. And there's a lot of other different enzymes. You have one called catalase that helps in the breakdown of hydrogen peroxide. A lot of your enzymes end in that suffix, ase. And they're always specific to a reaction, because one enzyme can essentially catalyze only one chemical reaction. Now, it can do it over and over and over again. Remember, they're not used up, but they are specific to a reaction. All right, so there's your protein summary there. Feel free to fill that in on your handouts. And then we're going to cover our last one, which is nucleic acids, okay? Now, nucleic means that they are in the nucleus, all right? Or they can be found in the nucleus, and then they are acidic. But once again, we have polymers, and remember that polymers are made up of smaller repeat units, and in this case, the monomer is called a nucleotide. Now, a single nucleotide has 3 parts. It has a 5-carbon sugar, okay, so you see the little pentagon here. That's because there's 5 carbons. So it has a sugar, okay, that sugar by itself would be a carb. There's a phosphate group. A lot of times it's just represented by a little circle, or maybe it has a P in it for phosphorus. And then there's a base, a nitrogen base, okay? So here's what they look like in their chemical structure. A lot of times we just use little symbols. But the nitrogen base is attached here, so we can see that we have a sugar, a phosphate, and a nitrogen base. And those 3 together make up the nucleotide. So that is the monomer for nucleic acids. And you've probably heard of this guy here, DNA. DNA is probably the main example of nucleic acids. And if you notice here, it ends in Na, and that stands for nucleic acid. Now the sugar in DNA is called deoxyribose, and that's where it gets its name, deoxyribonucleic acid. So that's how we get the DNA. Now another important nucleic acid is RNA. We'll talk about that in our biology unit. And so RNA has a sugar called ribose. So DNA has deoxyribose, RNA has ribose, and so deoxyribonucleic acid is DNA, ribonucleic acid is RNA. And these two molecules are very important because they carry our genetic information. The DNA has our genes, and then the RNA is very important in building our proteins, and so they have our genetic instruction. They essentially tell us who we are and kind of regulate our characteristics and all kinds of functions inside of our cells. So we're going to take a look at how they vary from each other, and so let's start with DNA. So DNA is found in the nucleus, and the DNA cannot leave the nucleus. It's too big to leave, and so the DNA is what holds our genes, and that's just a particular piece of the DNA, and the genes code for our proteins. Now DNA is made up of two strands of nucleotides, and they twist, so you can see that there's a strand here and there's a strand there. Okay, there's a strand. A lot of times you can think of it like a ladder. Okay, you have the sides of the ladder and then the rungs of the ladder. And so there's two strands of nucleotides. Okay, if you look closely, you can see that they're going in opposite directions. But there's two strands of nucleotides, and they twist to form a shape called a double helix. So it's kind of like a spiral staircase, like a twisted ladder. Now, one of the differences between DNA and RNA is the sugar. DNA has deoxyribose, which is where the D comes from, and then the sugars join up with the phosphates to form the backbone of the molecule. It's kind of like the sides of the ladder. It's kind of like the basic structure of the ladder. And then DNA contains nitrogen bases. And in the case of DNA, there are 4, A, T, C, and G, and they stand for adenine, thymine, cytosine, and guanine. And if you look on the inside of the DNA, the bases kind of pair up. A always pairs with T, and they're held with hydrogen bonds, those weak chemical bonds. And then C pairs with G, and once again we have some hydrogen bonds, those weak chemical bonds. And so that's just the basic structure of the DNA molecule. And so it just forms these long strands, this twisted ladder. The sugars and phosphates go on the outside. The nitrogen bases pair up on the inside. And that's going to be important when it comes to making copies of our DNA. And we'll talk about that later. All right, so there's 3 differences between DNA and RNA. DNA has 2 strands. It's a double helix. It has deoxyribose, and these are the bases that you find in DNA. Now, RNA is pretty similar. It's still made up of nucleotides, but in this case, we're going to have 3 different types. We have mRNA, which is a messenger. We have tRNA, which transfers amino acids. And we have rRNA, which is found in the ribosomes. And so RNA is a little bit smaller because it's single-stranded. So there's one difference between RNA and DNA. There's only one strand of nucleotides, and so RNA can actually leave the nucleus, where DNA can't. RNA is able to leave the nucleus, so you can find it all throughout the cell. It's not just isolated in the nucleus. And so it's one chain of nucleotides. It has ribose sugar instead of deoxyribose. Once again, that's where the R comes from. And then it also has 4 bases, but there is a key difference. So we still have an A, we still have a C, and we still have a G, but notice there are no T's, okay? Instead of thymine, RNA has a base called uracil. It's very similar to thymine, but there are no T's in RNA. Okay, so if it has T's, it's going to be DNA. If it has U's, it's going to be RNA. And so just a quick side-by-side comparison. We have double-stranded versus single-stranded, deoxyribose versus ribose, thymine versus uracil. And those are the 3 main differences between DNA and RNA. But both of them are polymers of nucleotides. Both of them carry genetic instruction. And that's going to take us to our final summary. So be sure you maybe want to fill that out. And then we've got just a little bit of review here. Now there are always lots of videos available on the internet. You can Google any topic that we cover in here, and I guarantee you, you're going to find videos. So there are 3 here that have been linked, but even if I don't give you videos, this is something you can always do. You can always look up videos for any topic. Khan Academy is always really good at breaking things down. Now this one, this is from my high school experience, but amoeba sisters are used a lot for basic biology. And so for these first couple of units, they might be handy. They're going to have lots of good videos. They are a little cartoon, but a lot of times they break it down into very easy to understand ways, and so you might want to check that out. They have a whole video about macromolecules. It's linked here. Another good resource is Crash Course. This guy's kind of goofy, but he's got lots of good videos. This particular video doesn't include the nucleic acids, though, because there's kind of its own video about the DNA and RNA and genetic construction. And even aside from these, there's lots of videos out there available. So if you just Google macromolecule videos, you're going to find lots of possibilities. So if you need some help, I would encourage you to check some of those out. Here's just a final summary chart. Carbs are sugars that give us energy. They're made up of monosaccharides that form di and polysaccharides. Lipids are fats and oils that don't like water. They have fatty acids, or they could be steroids. And the main thing is they're all hydrophobic, and they are not a polymer. Proteins are polymers made up of amino acids. Lots of different structures and functions. Remember, they form a chain, they twist and fold. They could be enzymes that regulate reactions. They could be structures like in our muscles. They could transport, they can fight disease. And then nucleic acids are like DNA and RNA. They're made up of nucleotides, and they carry our genetic instruction. You also have a little summary here. I think a great thing to do would be to take this and kind of add more information to it, so to kind of help you study. Maybe describe a phospholipid or draw a phospholipid, things like that. And don't forget, you do have a quiz over this chapter as well as your first lecture test. So please be preparing, and as always, if you have questions, please ask them. Okay, you're welcome to go see a tutor. You're also welcome to come and see me. So be sure you get help if you need it. And that's going to be all for Chapter 2.